Okay, class, now we're going to talk about, we talked about oxygen carrying in the blood. Now we're going to talk about carbon dioxide. Discussion of carbon dioxide transport. Okay, here's kind of the difference here. Let's look at it. Once carbon dioxide enters the bloodstream, it is carried in three forms. Now, first of all, we're going to pick up carbon dioxide down here at the bottom. See, in other words, we picked up, let me come over here. We picked up oxygen because it came in through the lungs. Okay, we picked up oxygen there. But I'm going to pick up carbon dioxide down here. So I'm going to start with internal respiration because that's where CO2 is produced. CO2 is produced in the Krebs cycle in the mitochondria in these cells and exits. So we're starting here with the loading of CO2. We're starting here with the loading of CO2. Okay? So we're changing the scenario because of where CO2 is formed. All right. So we come back here, and I'm going to say, once carbon dioxide enters the bloodstream, and is entering the bloodstream as we showed below, it's carried in three forms. Remember, O2 was carried in two forms. Some hooked to hemoglobin and some dissolved. But this is carried in three forms. Okay, 7 to 10% of CO2 is dissolved in the plasma. And again, we're back to Henry's law. How much can dissolve in the plasma depends on the partial pressure and so forth. Now, only 2 to 3% of O2 was dissolved in the plasma. Why can more CO2 be dissolved? Because it has a better solubility coefficient. Remember, CO2 had the best solubility coefficient of everything we named. Okay? Some is chemically bound to hemoglobin, just like oxygen was. Here's where the difference is, though. It is carried on amino acids in globin instead of iron and heme. So CO2 is carried on globin. And that's why they call it carbaminohemoglobin. Carbaminohemoglobin. Carbaminohemoglobin, because it is carried on that, on the globin. Now, when we look at that, I'm going to come down here and come back. CO2 is not transferred on heme. It binds to the terminal amino groups by forming carbamates. So remember, the amino acid had this aura group, and then it had this amine group. So it's hooking right here. You see where it's cooking? It's hooking right here to the amine group, right there. All right. So it's carried on globin instead of heme. Let's come back here. 70% is carried in a form we never saw with oxygen. So and that's called the bicarbonate form. That's going to be primarily in the plasma. It's going to form this bicarbonate. What's about that? Though carbon dioxide is a waste gas of the human, it must be transported to the lungs from tissue cells where it is a byproduct of the Krebs cycle in their production of ATP. But the human body takes advantage of the waste CO2 while it is being carried to the lungs. So it takes advantage of a waste. The CO2 will combine with water in the blood to make bicarbonate, and the key to this is to act as a pH buffer. We'll be talking about acid-base balance, but a pH buffer controls the pH in the blood and the body. A pH buffer. So you're taking advantage of this waste. All right? Now, so we say then that we have three pockets that this CO2 is in. Some is dissolved in the plasma, some is hooked to proteins, and then some is in the bicarbonate form. Okay, and we'll be looking at that and you'll be studying on that. All right, so we said CO2 is made in the Krebs cycle as a byproduct. Okay, CO2 has a good solubility coefficient. Oxygen had a 0.024, but carbon dioxide a 0.57. That's why it can dissolve so well in the plasma. Okay, we come here. We just talked about this. Okay. So now we come here and we see this particular aspect. Okay. Now, I'm going to show you how this is going to work. Okay. So what will happen is, first of all, I'll use this picture. From the tissue cell, CO2 will get into the interstitial fluid. Then from the interstitial fluid, it will go into the bloodstream. Okay. Now, here it's about a 45. In the blood, so in other words, we're coming, so this blood is coming, let's say, from the, so I'm going to come back here if I can find that. Let's see if I did something here. All right, so I'm going to go to this picture here. Now, CO2 coming from the arterial end of the capillary, that means coming from the lung, is 40. Here's a tissue cell. The tissue cell is a 45 because remember it's made in the cells as a byproduct. So if I come here, then I'm going to push a 45 into a 40. I'm going to push a 45 into a 40. When I push that 45 into a 40, then I leave and it's a 45 here going back to the heart. So let's go up here to look at it. So here's the tissue cell. The tissue cell had it as a 45. I'm going to come into here, coming from the tissue cell, and put it as a 45. Okay, I'm on that. Now I'm in that venous side, I'm coming, and then I'm going to go into the heart with a 45. But you see here, I'm a 45, but coming from here, I'm a 40. So I go from a 45 partial pressure to a 40. So that's what's happening here. So now with that in mind, I'll come here and use this one while you see that. You can see I'm pushing because I'm coming from a 45 into the blood as a 40. All right, now what's going to happen? All right, several things will happen. Some of the CO2 is going to push into the red blood cell. Some of the CO2 is going to dissolve in the plasma of the blood. Okay. Now, CO2, carbon dioxide, when it mixes with water, carbon dioxide, when it mixes with water, will non-enzymatically, no enzyme here, it's a spontaneous reaction, form H2CO3. Now, I want you to study this. H2CO3 is bicarbonate. Okay. Bicarbonate. Okay, actually it's called carbonic acid. Let me make a difference there. This H2CO3 is called carbonic acid. Carbonic acid. So CO2 plus water non-enzymatically can form this right here, carbonic acid. Then an acid, which we're going to see when we do acid-base, an acid, to prove it's an acid, dissociates, breaks apart. See, an acid is a proton donor. We're going to talk about that in the acid-base section in this chapter that I'm going to do. So right here is carbonic acid. It gets in there. It proves it's an acid by dissociating, which means to lose a proton, an H+. See that H+? Okay. Now what I have is bicarbonate anion complex. That negative side, bicarbonate anion complex. So you came in here, but it's slow because in the plasma there's no enzymes to do what I'm finna to show you. So let's see what we got. Some CO2 just dissolves. Some CO2 gets into this water in the blood, and through a non-enzymatic does this. Instead of dissolving, forms carbonic acid, dissociates, forms the bicarbonate anion complex, and here's the H+. Now the H+, so that you don't mess up your acid base, will bind to some of the plasma proteins. Remember the plasma proteins, albumin, globulin, so forth, fibrinogen. All right. Now I'm going to come down here. The majority, that's why this is so thick, the majority of this, see, this is a little thicker than this. That means only a little bit is doing this. more is doing this, but a lot more is doing this. Okay? Now, but before I get to the lot more, I'm going to go here. I'm going to go here. Okay? So we have, this is the least, this reaction here, in here. This, I would say, is like second least, this right here. Because see, this here would represent that 7 to 10%. This would represent that 7 to 10%. This would represent that 70%. That 70%. Let's come back here. But only a minuscule of the 70. See, 70% is bicarbonate. But this way of forming bicarbonate is the minority way. All right. So the CO2 comes in here and hooks to the amino acids of what? Globin, carbaminoglobin. Remember, the amino acids of globin, just like this. Now, what is that? That's that 20 to 23% right here. But the 70% is really this. And of that 70%, a little bit did it this way, but the lion's share is going to do it this way. Now, let me explain what's going to happen. Inside the red blood cell, genes that have done this make an enzyme called carbonic anhydrase. Carbonic anhydrase is an enzyme that can speed this reaction up a lot more than without the enzyme. You know what enzymes do? Speed up reactions. So this reaction inside the red blood cell occurs. When that occurs, then you form again this carbonic acid. The carbonic acid proves it's an acid. Okay? Proves it's an acid. Now, what I now have is this, and see, this is a proton acceptor. So the bicarbonate anion, when we get to the chemistry part, this is a base. A base likes to accept a proton. This is an acid right here, H+. So what I need to do is, so that I don't mess up the pH up in here, I need to get rid of you. Okay, I need to shuttle you out. So what's going to happen, inside the red blood cell, the cell membrane has a special, what they call antiporter, we'll call it. The bicarbonate gets shuttled out, so it doesn't mess up the acid base, because remember, this is a base. But if I take a negative out, because I don't want to mess up bulk electron neutrality, go back and look at that in my ANP1 if you don't recall, or didn't get it, if you didn't take me. So I'm going to shuttle this out. If I shuttle that out, so I don't mess up the bulk electron neutrality of charges, I need to bring something from the outside that likes to come in. Chloride is an ion in higher concentration outside a cell than inside a cell. So the chloride comes in. So this goes out, chloride comes in. Okay, and that's fine in there. Now, when this goes out, some of it may hook to sodium. Remember, sodium is an ion in higher concentration outside a cell and forms something called sodium bicarbonate. Now, all of this is happening in the loading aspect. All of this is happening in the loading aspect. All right. Now, what about this right here? This part right here will hook to some of the, see this, that's a positive. It'll hook to some of the basic amino acids in globin. See, H+. So I'll get rid of the H+ by hooking, because H+ is an acid. So I'll hook to some of the basic amino acids in globin. That gets rid of this. So I get rid of the H+ by hooking to that, and I get rid of this by shifting out and bringing you in. So if everything's set, I'm loaded in. Some in the dissolved, some did it here, but no enzyme. A lot made bicarbonate here, you see. And then some CO2 binds to globin, as I showed you before in the amine group. Okay? So we have everything loaded now. We have everything loaded, and I'm going to close this one, and then we're going